now we're going to move away a little bit from the biotechnology side of things, and we're going to focus really on how DNA and genes specifically can move throughout particular populations and gene pools. So let's define a few key terms first. So firstly, population. This is a group of organisms, so living things of the same species that live in a particular place at a particular time. Secondly, a gene pool is the sum of all alleles carried by members of a population. So not one individual, but every member of that population. The allele frequency is a term used to denote how common each allele of a particular gene is within a gene pool in comparison to others, and gene flow refers to the transfer of alleles from one population to another, and that largely occurs through the process of migration. So, we've previously seen this slide before. Changes to allele frequencies within the gene pool are due to mutations, which we've looked at. However, secondly, natural selection, third, gene flow, and fourth, genetic drift. Now, as you can tell, our focus today is going to be on gene flow. So, a very quick recap for us. We've looked at mutations previously, and we've seen that mutations cause permanent changes to the DNA of individuals. Now, mutations are the only process capable of creating new alleles or genes, and as a consequence of that, the only process capable of creating variation, okay? Specifically, genotypic and phenotypic variation. So mutations effectively introduce new alleles, and they can introduce alleles which end up being favourable for the survival of particular species. So for instance, over on the left here, we have the white-bodied and the black-bodied peppered moth. During the Industrial Revolution, when trees were covered with soot, the black-bodied pepper moth had a survival advantage because it was no longer as easily seen as the white-bodied peppered moth. So when predators such as owls were looking at trees, for instance, they could clearly see the white body of the peppered moth compared to the black-bodied pepper moth, and this led to a change in the allele frequency between these two species, or between these two different variations of the allele for body colour in these peppered moths. So if a favourable allele is passed on to offspring, this will only take place in germline cells, okay? So it won't occur in somatic cells, and gradually, if it's passed on to offspring, and in this case the white-bodied peppered moth is eaten before it's able to reproduce, the other members of the species will increase in number due to that process known as natural selection, which we'll dive into a little bit later on. So some mutations can be selectively advantageous, okay? And this is where they only provide particular advantage under very limited circumstances or conditions. So now that we've discussed this idea of mutation, what we want to look at is this idea of migration, so relating it to gene flow now. And when we're talking about gene flow, we can't have it without migration. So migration, very simply, is where organisms move to a new area, and we can have two different types. So we can have immigration. So this is effectively what I'll highlight here in green, is this portion where we have an individual moving into a population. And on the other side of things, we can have emigration, which is the movement of individuals out of a population. So in the case of migration, it's impossible to have one without the other. So in order for an individual to immigrate into a new population, they have to emigrate from a previous population. So what is gene flow itself? Again, here's our definition at the top. Let me highlight it in full to be really clear. Gene flow is the movement and exchange of alleles between populations of a species. So this can't occur without migration, okay? So with that migration process, immigrants are going to introduce new alleles, and you know what, we'll do the same colour as I did on the previous slide. So immigrants will introduce new alleles, and emigrants are going to remove alleles from the original population. Now, if a trait isn't that frequent in the original population, it can become more frequent if an immigrant to the population brings that particular allele with it, that particular gene with it, and as a consequence, these allele frequencies, the frequency of particular traits, can change over time. Now, what occurs commonly with gene flow is that it's going to reduce the differences between the gene pool of the source and the recipient populations as these genes migrate from one population to another. And what happens is that the allele frequencies between the two populations, being the original and the destination population, generally over time will become more similar. So, what's going to stop gene flow? Well, we know that it's promoted by migration. However, migration can be inhibited in two significant ways. Firstly, and most commonly, geographical barriers. So we're talking things like oceans and rivers. We're talking mountain ranges, deserts, big ice sheets. All of these things can prevent species from migrating from one population to another. And also in human populations, we can also have socio-cultural barriers. So things like economic status of individuals, educational background, their particular religious beliefs, and even language. Okay, so when it comes to population isolation, generally speaking, this is going to result in an increase in the rate of inbreeding, which is, as we'll define it here, mating between closely related individuals. Now, what this does is it increases the chances of individuals with the same particular recessive alleles being expressed in their offspring. And unfortunately, this is going to result in a reduction in genetic diversity. So, for instance, we can have an example with geographic isolation here within Australia. So Australian inhabitants isolated for thousands and thousands of years after the Ice Age as sea levels rose, creating the island that is Australia. And we can also have socio-cultural isolation, and we'll look at some examples of this a bit later on, but where certain religions only allow from marriage and breeding within members of their own community. So an example of a small population group is the Vedoma people within Zimbabwe, and they're sometimes referred to as the ostrich foot people of Vedoma. As we can see here, their feet have this unique congenital deformity where the number of toes is significantly reduced, and they resemble the feet more of ostrich than a traditional human foot, if you like. So this is an example of a small population with limited gene flow, and as a consequence, these recessive traits or particular recessive traits have a higher probability of being expressed, and it's reduced the genetic diversity of that population.